developing here in the Institute of Biomedical Sciences. Live stream. So the meeting is being live streamed.
Sorry.
You're good? Okay. So I'm going to talk to you a little bit about my research project that is entitled Host-Directed Therapies for TB, Targeting Iron Homeostasis, Immunometabolism, and Lung Inflammation. So I try to make a summary of what I have done to start this project and what I have been doing these last few years. So I'll try not to take too long because we're already a little bit late. So this project, as Gustavo told you, was started in Ribeirão Preto Medical School, where I was a young investigator from FAPESP for roughly 3 years. And then now I moved to Institute of Biomedical Sciences, where I'm going to continue these studies. So just to talk a little bit about tuberculosis. Tuberculosis is an infectious disease caused by this bacterium here, Mycobacterium tuberculosis. It affects mainly the lungs, so it's transmitted via air. So an infected individual will cough or expel droplets of saliva or sputum containing live bacteria that can then penetrate the lungs of a healthy individual and cause disease in the lungs. Although TB can also spread to other organs, and you can have TB in the brain, like in the meningeal TB, bones, lymph nodes. There are several tissues that can be infected by bacteria, but the most common is the pulmonary TB. And TB has been one of the most deadly diseases for mankind. So just in the last 2 centuries, the estimates are that TB caused roughly 1 billion deaths. So this is too much, and TB continues to be a huge public health problem nowadays. So this is the last World Health Organization TB report, and this is showing that in 2021, this is from 2022, but it brings data from 2021. There were around 1.6 million people that died from TB, and 10.6 million new cases of TB were registered. And as you can see here, this is the timeline of TB cases that have been registered from 2000 to 2021. There was a minor increase in the number of registered cases, and this happened because of the COVID-19 pandemics. So there was a huge major drop in the notification of new cases, and with that also people stopped having access to treatment, to diagnostic treatment, and etc. So the number of new cases increased, and the number of deaths also increased. And why is TB still a major public health problem? We have a vaccine for TB. That's BCG. Almost everybody here must have gotten BCG vaccine when was a newborn. And BCG is really good to prevent the development of disseminated TB in newborns, but it's not so good to prevent the development of adult pulmonary TB. So maybe the memory T cells are not very long-lived. It's very probably, if we take a PPD test in everybody here, a lot of people won't respond, although everybody have been vaccinated with BCG. So it does not protect very well against pulmonary adult TB. But there is a treatment. There is an antibiotic treatment that is very effective, actually. So if the person takes this treatment correctly, around 90% of people will be cured from TB. But it's a very aggressive treatment. So it's 4 antibiotics that are administered every day for 2 months. And then there's a continuation phase of 4 months, also with 2 of these antibiotics. So a lot of side effects develop. A lot of people do not comply with the full treatment. So they abandon treatment before they are completely healed. So this promotes the development of drug-resistant bacteria, and these are way harder to treat. So today we have these multi-drug resistant strains that they are resistant to 1 or 2 of those first-line drugs, and then several other drugs that may be injected or already taken need to be administered. The common length of treatment that should be 6 months can be extended to up to 2 years. The success rate is way lower, it's 50%. Even so, the side effects will be even more severe. So a lot of people also drop this kind of treatment. then we can have the development of these extensively drug-resistant TBs, or they are resistant to almost all of those second-line drugs as well. Mostly just experimental drugs are working on these strains, but as you can imagine, the experimental drugs are going to be even more aggressive, so the side effects are even more severe, and the success rate drops a lot, 30%. The treatment will last at least two years, but it can take way longer. So because of that, one thing that can be done to help control TB is to develop new treatments, right? So novel treatments that can eliminate this disease more rapidly and more effectively, and do not rely exclusively on antibiotics. And that's what I have been trying to study. What are the host targets, the host biological processes that can be targeted for the development of new drugs? So we take biological processes from the host that favor disease development. Either they can be promoting pathogenesis, or they can be lowering immune responses. So we can study those targets, target them, and develop new treatment strategies for TB. So when I was in my postdoc, actually before starting telling about the results, what are the main targets for host-directed therapies in TB? So because for that, we need to understand a little bit, and I'm going to present very summarized here, how is the immune response to TB? So briefly, TB enters the alveoli, and they are going to encounter mainly alveolar macrophages. They are going to be the cells that are going to phagocytose those bacteria, and the bacteria have developed several ways through evolution to avoid the effector response of these macrophages. Although TB has several ligands that are going to trigger innate immune receptors, and that are going to induce the production of TNF and IL-beta mainly, that are important to induce the maturation of phagosomes and elimination of the bacteria. These bugs have evolved ways to avoid phagosome maturation and acidification, as well as the phagolysosome fusion. So by doing this, they can escape the effector mechanisms of macrophages, and then you're going to need an adaptive response to be developed to control this bacteria. So mainly because of these ligands that TB has on its surface, it can induce the production of IL-12 by dendritic cells. And this IL-12 is going to be important for the differentiation of Th1 lymphocytes that are going to produce interferon gamma and TNF alpha mainly. That are very important for inducing the production of reactive oxygen species and nitric oxide and reactive nitrogen species that are going to be important for controlling the bacterial replication, for the macrophages to control bacterial replication. Although, even though many of these bugs do not die, so the host developed a way of controlling the spread of this bacteria to other organs. So a classical inflammatory response develops that is called formation of granulomas. And in TB granulomas, the infected cells are contained in the center of the granuloma, and these are going to be surrounded by macrophages that differentiate into epithelioid cells, and also a ring of lymphocytes that are going to be actively producing pro-inflammatory cytokines to try to activate those infected cells and lead to the elimination of bacteria. Several times, these granulomas can be very successful, and you have the growth of fibroblasts around it, so you have fibrosis, so around these granulomas, and this can actually control control the dissemination of the bacteria, and these calcified granulomas will contain bacteria. But many times these bacteria continue to replicate and kill those infected cells. This is going to generate a necrosis. It's called caseous necrosis because it resembles cheese, and these granulomas can burst, and this is going to lead to the transmission of the bacteria if these fourth granulomas reach a bronchic branch, and this bacteria can be expelled. So what happens for the bacteria to induce necrosis is usually an exacerbation of the inflammatory responses. So you have that environment in the granulomas, where you have active production of pro-inflammatory cytokines. And these bacteria, when they escape to the cytosol, they can lead to a huge production of IL-1 but not just IL-1 When the production of interferon gamma TNF by Th1 cells and also TNF by the own macrophages is too high, this can lead to a huge production of ROS and induce these infected macrophages to die by necrosis. Infected macrophages die by necrosis. This is going to favor the dissemination of bacteria and is going to promote more pathogens. So this is what happens when this necrosis occurs. So we're going to have destruction of the lung tissue. So it's not just an infectious disease, it's actually an inflammatory disease, because the inflammation is what actually leads to the destruction of the organ. So what can be done in terms of host-directed therapy to control TB? Many strategies are focused on increasing immune response to the bacteria, to try to make these macrophages to eliminate the bacteria more quickly and more effectively. Or on the other side, there are anti-inflammatory drugs that are administered together with the antibiotic treatment, so you can lower this inflammation and have a better control of tissue damage. On the other side, also granulomas, when they're formed and there is the deposition of collagen, for example, these can impair the penetration of drugs. So there are some strategies that are focusing on disrupting the granuloma so the drugs can penetrate and eliminate the bacteria. So now I'm going to start to tell you a little bit of what I have been doing in these last years. So when I was in my postdoc, I was studying the role of this enzyme here, hemoxygenase 1. So this is an enzyme that degrades heme molecules, and it releases equimolar amounts of biliverdin, carbon monoxide, and iron. So the expression of HO1 is the inflammatory isoform of the enzyme, is induced in response to TB infection in the lungs of several different hosts. So we have induction of TB of HO1 in the serum and in the lungs of humans with TB, monkeys with TB, and I didn't bring here, but also in mice and in rabbits, you have induction of HO1 production. And so I wanted to study what is the role of HO1 during TB infection. And for that, we used a pharmacological approach. So we treated mice with this team protoporphyrin 9. This is a HO1 inhibitor. So it resembles heme, that is the natural substrate for hemoxygenase, but instead of iron, it has a team center here. And what happened when we treated mice infected with TB? For just 2 weeks here, we had a major reduction in the bacterial loads in the lungs of these mice. And when we combine this with antibiotic treatments, here 3 weeks post-treatment, we have a reduction in bacterial load just with oxygenase inhibitor that is similar to what the antibiotics do. Here we treated with 3 of those antibiotics. And when we combine it, we have an even more robust reduction. And when we did this long-term, we had the elimination of bacteria in the lungs of these mice in around 4 months, something that should take 6 months normally, just with the drugs. So we saved 2 months of treatment in the mice, right? So then we were interested in studying how does this work. So just in a regular experiment that was trying to mimic what would happen in an HO1 knockout mouse, I said, oh, let's treat this mice since day 0, right, with the inhibitor and see what happens. So we treated this mice since day 0 with HO1 inhibitor or not. And the interesting thing is that in these 3 first weeks of treatment, nothing happened. So the bacterial load was normal. But when we continued from 3 weeks on, then we had a reduction in bacterial load. And it's 1 log reduction. This is a lot for only 3 weeks. So we started to think, well, one thing that is very characteristic in TB is that the T cell response, it takes a long time to develop. So if you infect a mouse or a human with TB, the T cell response will only be detected in the lungs after 3-4 weeks of infection. So curiously, we only started to see an effect of HO1 inhibition after the 3rd week of infection, right? So as you can see here, this is the kinetics of interferon gamma expression in the lungs. You see it's only detected 3 weeks post-infection. The CD4 T cells also start to express TBET on the 3rd week post-infection. So when we treated mice that lack T cells, that are T cell deficient with this inhibitor, we saw here the nice 1 log reduction in bacterial load in wild-type mice, but this did not occur in mice that don't have T cells. And the same occurred if these mice did not have interferon gamma, or if these mice did not have the expression of INOS. So the effect of H1 inhibition in inducing and decreasing bacterial loads is dependent on T cells, T cell dependent production of TNF or of interferon gamma and expression of inos, which occurs in infected macrophages. So we moved on to start to understand what were the mechanisms that were governing that. And when we infected bone marrow-derived macrophages that were wild type, when we treated those cells with interferon gamma, we had a nice reduction of bacterial load. And when we treated with interferon gamma plus inhibition of HO1, the reduction was even higher. But if these cells do not express inos, then I don't have this potentiation of the interferon gamma induced microbacterial killing. And so we started to look if really the treatment with the inhibitor of H1 would induce a higher production of nitric oxide and expression of inos. And indeed, those cells that had H1 inhibited expressed more inos and produced more nitric oxide. And then we started to wonder, how could this be happening? So HO1 degrades heme and produces three products, right? Diverting carbon monoxide and iron. So we wondered, and if we give back those products to the infected macrophages that were treated with the inhibitor, what will happen? So here we have this nice effect, right? I didn't put non-treated here, but the bacterial loads are higher. So this is just interferon gamma and interferon gamma plus the inhibitor. If we give back carbon monoxide, nothing happens. If we give back biliverdin, nothing happens. If we give back iron, we revert the effect. So the iron that's being released from these heme molecules is impairing the effector activity of these macrophages that is being induced by interferon gamma and depends on nitric oxide. And indeed, when we treated these macrophages with HO1 inhibitor, as you can see here, there is an increase in the expression of inos, but when we put iron back, this goes down. As you can see here, this is SNPX, that is the inhibitor, this is SNPX plus iron. So the expression of inos goes down. And when we do the contrary, we treat the cells with gamma plus SNPX, there is an increase in iron, but when we treat these cells with an iron chelator agent, so it's gonna sequester all the iron that is there, the induction of ions is huge. So we saw that iron, the intercellular iron pool, is some way controlling the induction of ions expression in the infected cells. And indeed, when we took lungs of mice that were infected with TB and treated or not with H1 inhibitor, here we did a staining for iron. So we stained these lungs with burst staining, and then we amplified this with the aminobenzidine. So you have in brown here, the spots denoting iron accumulation in the lungs. You can barely see that in mice that were treated with the H1 inhibitor. So not only in vitro, but also in vivo, inhibiting H1 is reducing the accumulation of iron in those cells, and somehow this is helping the macrophages to work better for controlling bacterial replication when they're activated with interferon gamma. So this was the conclusion of this paper, right? So we saw when HO1 is active, you have a lot of iron. This is going to somehow inhibit inos expression when the cells are activated with interferon gamma, and you can have happy bacteria proliferating inside these macrophages. But if I block HO1, this is going to lead to an upregulation of inos, more production of nitric oxide, more toxicity against the bacteria. So with that, I developed my young investigator project that I submitted to FAPESP and started to develop it. I came back to Brazil. And before that, there's also the work of an important previous member here from Institute of Biomedical Science, Edu. I worked a lot with him in NIH, and Edu also saw that these macrophages, when you have accumulation of iron, you have induction of necrotic death by ferroptosis. When he blocks the induction of ferroptosis, you have a nice, huge reduction in inflammatory reaction and also in the bacterial load. So with this, we saw that iron can be both impairing the microbicidal effects of macrophages, and the accumulation can also induce necrotic cell death that's going to promote bacterial dissemination and pathogenesis. So my goal was to study how the modulation of iron homeostasis happens in TB, what's the role of this modulation, and if there are molecular targets for development of new treatments that we can use to help the antibiotic treatment to be more effective. So a macrophage acquires iron through several ways. One very common way is by phagocytosing senescent red blood cells. And so these red blood cells are going to be degraded, heme is going to be released, this is going to be degraded by HO1, and iron is going to be released. Heme and hemoglobin can also be acquired from the serum. They're going to suffer the same fate, but they can also internalize iron from transferring molecules from the circulation, and this is going to be endocytosed. Iron, ferric iron is going to be metabolized through ferrous iron, and this is going to be transported to the cytoplasm by DMT1, that is this transporter here, that also imports iron from the outside because it's also expressed in the cellular membrane. And iron will be either stored in ferritin molecules. The free iron can induce production of a ROS by the Phantom reaction, but it can only be exported by this molecule here, this is ferroportin. So there's only one exporter of iron atoms from the cell, right? So the regulation of ferroportin is intimately related with the regulation of inflammation. So its the main mechanism that the body uses to regulate ferroportin expression in macrophages and in all cells is by the production of this hormone here, hepcidin. So hepcidin is going to bind to ferroportin and induce its internalization and lysosomal degradation. And what's interesting about TB is that almost all the ligands and almost all the inflammatory mediators that promote degradation of ferroportin are produced during TB infection. So we have IL-22 and IL-6 that can induce the production of hepcidin. We have triggering of toll-like receptor 4 that can induce the production of hepcidin. And on the other side, it was also described that the triggering of toll-like receptor 2 can induce a downregulation of ferroportin expression, gene expression. So TB has all this. So maybe during the infection, we can have what may happen is a downregulation of ferroportin, and this can help promote suppression of macrophage effector activities and also promote necrosis by ferroptosis. So the work by Marina is a master's, no, she just finished her master's degree, is going to start PhD here now. She started to study what's the role of ferroportin in macrophages during TB infection. So for that, we first need to synthesize hepcidin and see if it worked for inducing the downregulation of ferroportin. So we were willing to test if treating the macrophages with hepcidin could impair their microbacillary effect. So here is just a validation. We have macrophages here that penetrate. It's hard to see here, but there is some light staining in red that is the ferroportin. When we give iron, we induce expression of ferroportin, you can see here. In hepcidin, we can barely see anything here, but we cannot see very well here, but we can see here. So by flow cytometry, we have here in pink is the expression of ferroportin control cells. When you treat with hepcidin, there is a decrease. When you treat with iron, there is an increase. So she tried to treat these cells with hepcidin, but nothing happened. So we thought, well, black 6 cells, normal cells, they must be upregulating again the expression of hepcidin. We may be losing the effect here. So let's try to do some knockouts. So before that, actually, she characterized that macrophages could have the expression of hepcidin modulated. So this is just bone marrow derived macrophages from black 6 that she did not infect or infected with TB in different multiplicities of infection. And she treated these cells with hepcidin, did not treat, just infected, or treated with iron, that is an inducer. So she saw that in macrophages that are supplemented with iron, the infection has a major effect in inducing high expression of ferroportin. And in vivo, also by infecting black 6 mice with a reporter bacteria, so a reporter bacterium, so this is MTB that expresses mTERI, we could analyze infected and non-infected. This is alveolar macrophages, and we see that the infected cells express higher amounts of ferroportin. And also when we take the interstitial or the parenchymal macrophages and divide between the infected and the non-infected cells, we also see a higher expression of ferroportin in infected cells. So actually it seems that TB might be inducing the expression of ferroportin. So then we were wondering what happens if we knock out ferroportin. So ferroportin deletion is lethal. So we had to work with ferroportin floxed mice. She spent a huge time trying to make this model. We got these ferroportin floxed mice in collaboration with Nancy Andrews and David Fraser. And we crossed these mice. We ended up crossing it with Lysampre because she spent a huge time trying to cross with CSFR1 pre. And turns out it was lethal. So CSFR1 pre must be expressed in some other cell that not just myeloid cells. But with Lysampre promoted, it worked. And when Marina infected those macrophages in vitro from wild type cells or from knockout cells, without any kind of supplementation, they were more susceptible to bacterial replication. This is 4 days after infection with an MOF1. And if she puts iron, like these are two separate experiments, so the numbers here are not exactly similar, but like we have here 2 times increase in bacterial loads, we have here almost 3.5 increase in bacterial loads when you supplement iron. And although I did not bring here, this was correlated with higher cell death when the cells do not have ferroportin and they are supplemented with iron. So we suspect that the main thing that's happening here is through induction of fructosis, although she's going to work more deeply in that on that sense right now. But although our preliminary results point to the fact that the induction of ferroportin expression during the infection with mycobacterium tuberculosis is protective for the host. And now we want to evaluate which are the mechanisms that are involved in this induction. So it's very well characterized what decreases ferroportin expression. It's not very well characterized what induces it. So this is going to be part of Marina's PhD thesis. And we are betting on a transcription factor that's called SPIC. It's very important for B cell biology, but it's a powerful inducer of ferroportin expression. So I'm running after these mice now. They're only available in Japan. I need to buy them and bring them home. So we also started working on how iron homeostasis in lymphocytes can modulate the immune responses to TB. So it's not very clear what iron does to Th1 cells. There are reports showing that if you take iron out of those Th1 lymphocytes, they're going to stop expressing interferon gamma. But there are other reports saying that if you put iron, that you're going to prevent them from expressing TBAD. So it's not very clear. So actually, it's unknown what how the iron homeostasis of T lymphocytes can modulate not just Th1 differentiation, but we're going to use TB as a model here that's very dependent on Th1 responses to try to study how iron homeostasis affects the biology of T cells. And lymphocytes specifically rely mostly on internalization of transferrin bound iron. And this happens by the binding of this transferrin to CD71, that is the transferrin receptor. This is going to be internalized. And as I said, this ferric iron is going to be converted to ferrous iron and going to be transported into the cytosol by this DMT1 transporter. And it only can be exported by ferroportin. So Ana Clara is a postdoc that just started in my lab. So we have very preliminary results here. She just measured the expression of DMT1 and ferroportin in the surface of T cells during TB infection. And she saw that in the 4th week post-infection, when we have establishment of disease in the lungs of these mice, the activated cells, right, activated CD40 cells, CD44, or Th1 cells express higher amounts of both the importer and the exporter of iron. So we have here ferroportin being expressed most highly in CD44 positive cells and in TBAD positive cells. But DMT1, that is the importer, is also most frequent in CD44 positive cells and TBAD positive cells. So these are very preliminary data. Ana Clara is now working on generating the conditional knockouts. So we also got the DMT1 floxed mice, and we're breeding the ferroportin floxed mice and the DMT1 floxed mice with CD4 pre. So we're going to delete these transporters in T lymphocytes, and we're going to see what happens with infection. So she's doing some preliminary studies just in vitro in Black 6 now, treating with pharmacological blocking iron availability or giving iron and seeing what happens. But the mice are halfway ready. We should have some good results soon. And then we also wondered, how can iron be modulating the activity of these macrophages, right? Because I showed you the main results we had are in macrophages. Iron was impairing their effector responses, also inducing fructosis. But how does iron modulate the effector functions of macrophages? And this is where our project started to mix a little bit with immunometabolism, because then we started to turn our eyes to HIF1 alpha and HIF2 alpha. So these two hypoxia-induced effectors, their expression is induced in cells in response to hypoxia, right? And how does this happen? So actually, oxygen is needed for the enzyme that degrades the HIF1 and HIF2 alpha subunits. So it's used as a cofactor by this protein hydroxylase enzyme that's going to promote ubiquitination and degradation of HIF1 alpha. When you don't have oxygen, you have hypoxia, this enzyme is inactivated, HIF1 alpha is stabilized, it migrates to the nucleus, induces glycolytic metabolism and the production of several pro-inflammatory cytokines. But what happens is that iron can also be used as a cofactor by this prolyl hydroxylase. And so when you have a lot of iron, this enzyme is very active and can degrade HIF1 alpha. So it's shown that when you have a lot of iron, HIF1 alpha goes down. But when you have low iron amounts, then HIF1 alpha is stabilized and can migrate to the nucleus and induce metabolic changes and production of pro-inflammatory cytokines. And what happens with HIF2 alpha? That is the other subunit, the other isoform of HIF alpha that is not very well studied. So HIF2 alpha works on the contrary. High iron amounts induce the expression of HIF2 alpha, while low iron amounts induce the downregulation of HIF2 alpha expression. And how does that happen? That happens because the RNA molecules for HIF2 alpha, they have this iron responsive element, that is a DNA RNA motif, that is going to be targeted by this protein here, iron regulatory proteins. So when the iron amounts inside the cells, the iron levels inside the cells are low, these iron regulatory proteins, they have a conformation without binding to iron, that they are very, the affinity for these iron responsive elements is very high. And so when iron is low, IRP is going to bind to the IRE and is going to prevent HIF2 alpha from being, from the protein to be, the RNA to be translated into protein. When iron is high, iron is going to bind to IRPs, it's going to alter its conformation. It no longer is able to bind to the IRE from the mRNA from HIF2 alpha, and then this mRNA is released to be translated into protein. So for HIF2 alpha, high iron induces HIF2 alpha, how it decreases HIF1 alpha. And HIF1 alpha is very important for the immune response against TB, for host resistance to TB. But the role of HIF2 alpha is not known. So HIF1 alpha is very well characterized in M1 macrophages. It induces Warburg effect, that is the glycolysis in the presence of oxygen, induces production of IL-1 beta, TNF, INOS, all of those are important for resistance to TB. But HIF2 alpha, its role is not very well characterized, but it's a very powerful inducer of arginase 1 expression. Arginase 1 competes with INOS for arginase, and that leads to the polarization of macrophages to the M2 phenotype that is more important for tissue repair and scar formation and things like that. So our next question here was to evaluate if there's a role for HIF2 alpha in TB. So Josiana, that's a PhD student in my lab, started this project. So we got the HIF2 alpha flux mice. Also, HIF2 alpha knockout mice do not live very well because they have a huge anemia. HIF2 alpha is very important for erythropoiesis. But we crossed this with a promoter that's going to delete HIF2 alpha in myeloid cells to start evaluating what could be the role. of this transcription factor in TB. And when Josie cultured BMDMs, she saw and infected them with TB. Here we have in white the wild type cells, and in gray the HIF2 alpha knockout cells. So after 4 days of infection, the knockout macrophages are more resistant to TB replication. If she treats these cells with gamma, both respond well. But if the cells are just infected, they're more resistant if they don't have HIF2 alpha. She measured nitric oxide, iNOS, and arginase. She actually didn't see a good difference in the production of nitric oxide. Although there was a slight reduction here, it's significant, but it's very similar, the expression of iNOS, and no difference in arginase. But something that was interesting was that while the infection with TB induced the expression of HIF1 alpha in these macrophages, it did not change the expression of HIF2 alpha. But if she infected knockout, HIF2 alpha knockout cells and wild type cells, although here the plot's not very well, but not very good, but when she calculates the ratio, the expression of HIF1 alpha is higher in cells that do not express HIF2 alpha. So we're wondering how HIF2 alpha may be counter-regulating the expression of HIF1, and maybe this is what's making these macrophages more effective in killing MTB intracellularly. Josie also infected mice, the live mice with TB, and she saw that 7 weeks post-infection, there was a reduction in bacterial load. This was associated with an increased expression of iNOS in parenchymal macrophages, but also a higher expression of HIF1 alpha, at least the mRNA expression of HIF1 alpha in lung tissues. And then we said, let's treat mice with an inhibitor of HIF2 alpha dimerization and see what happens. So when she infected mice and treated these mice orally with an available HIF2 alpha inhibitor, there was a nice reduction of bacterial load. This was also only 3 weeks of treatment. So maybe we got a very strong candidate for a host-directed therapy here, and we're starting to work a little bit more on what's the mechanism that's happening here, and if we really can use this as an adjuvant therapy for TB, combining it with antibiotics. Well, this was the part that we're dealing with iron, but there's another part of the project that we're also dealing with lung inflammation. And in this part, we're examining the role of these TAM receptors. These TAM receptors, they're called TAM because there are 3 members in this family, tyrosine 3, exon, and MERTK, so it's the initial. These receptors are tyrosine coupled receptors, and what they do is they bind to these 2 ligands here, that's Gas6 and PROS1, and these ligands are going to recognize phosphatidylserine that is exposed in apoptotic cells. So these TAM receptors, they mediate efferocytosis, that is the phagocytosis of apoptotic cells. So this is what happens. Gas6 has affinity for all of the 3, PROS1 for only MERTK and tyrosine 3. And to start studying what's the role of these receptors in TB, and if they can be targeted for a host-directed therapy, well, I forgot to say, but they have an anti-inflammatory action in the cells, right? So when cells have TAM receptors triggered, they're going to produce more IL-10, but on the other side, less TNF, less IL-1, less IL-18, less IL-6, as well as a lower expression of CD86, MHC class 2, so more accessory molecules to induce the activation of lymphocytes. So what he did, we contacted Carla Rotlin, and she provided us with the mice that are knockout for AXL and knockout for MERTK, and we started studying what could be the role of these receptors in TB. So Andre, that is now a PhD student in my lab, he started first to evaluate if TB infection could induce the expression of these receptors in vitro in bone marrow-derived macrophages. And he saw that very nicely AXL expression is strongly induced in response to infection. And this only happens when the bacteria are virulent, because if they lack Z6, that is a main, I think the major virulence factor from TB, you don't have that much induction of expression of AXL, but MERTK is highly expressed in the cells no matter what. But when he turned to evaluate in vivo in macrophages from the lungs, if TB infection induces the expression, he saw that mainly in this, we call it IV negative because we perform this IV staining in the mice before we sect them. So we inject CD45 intravenously. So in the lungs, it's a very irrigated organ. Even if you perfuse the lungs, a lot of cells keep bound to the vascular bed. So with this, we can select just the cells that were really infiltrated the lung parenchyma. And when we evaluate those cells, we have alveolar macrophages, IV negative or parenchymal neutrophils, and the IV negative mononuclear myeloid cells. Here, these are mainly CD11 positive, CD11B positive cells. These cells mainly have a huge upregulation in the expression of MERTK and AXL when the infection is established. So here you can see a naive mouse, an expression of MERTK in an infected mouse, and the expression of AXL in a naive mouse and an infected mouse. And when we infect those mice with a fluorescent bacterium, and André isolated here only the positive cells, we saw that most of the infected cells express both AXL and MERTK. So this tells us that AXL and MERTK may be acting in infected cells in vivo to do something. So then André asked West, well, and if we infect the knockout mice, what happens? For our surprise, both AXL knockout and MERTK knockout mice did not present any difference in bacterial loads in the lungs with 3 weeks post-infection, so a very acute phase, and also later with 3 months of infection. The numbers of myeloid cells did not change. The levels of iNOS expression did not change. And although there was a higher production of TNF and interferon gamma with 3 weeks post-infection, later on this fades, and there was no difference. So mice that are deficient in AXL or knockout, there's no effect in resistance to TB infection. But there is a very interesting thing that happens with these receptors. I don't know if you heard about ADAM17. It's a protein that cleaves a lot of receptors, cytokines, or whatever that is expressed in cell is expressed on cell membranes. So it's very important, for example, for the release of TNF that is expressed in cell membranes, IL-6 receptor. So you have soluble IL-6 receptor, you have the trimers, the TNF. ADAM17 has a very important role in cleaving it and releasing those to the environment. So ADAM17 can also cleave AXL and MERTK. And in this case, the soluble AXL and MERTK are going to bind Gas6 and FOLS1, also those that are expressed in the apoptotic cells, and it's going to prevent the action of the system, right? So it's like a decoy receptor. It's going to block. It competes with the receptors that are in the cells. So let's see if we have accumulation of the soluble receptors in the lungs. And what we saw is a huge accumulation of Gas6 in the lungs of infected mice, but also a huge accumulation of the soluble receptors. So maybe in a normal situation, you have a lot of expression of AXL and MERTK in the infected cells. You have ADAM17 acting, and they're being released, and maybe the system is not working very well. So if we give Gas6 to the mice, and we give more, will it work? So we infected Black 6 mice, and after 2 weeks of infection, we started treating these mice, or not, with Gas6 or with Eagle every 2 days for 2 weeks. And for our pleasant surprise, there was one log reduction in only 2 weeks of treatment. So maybe we have a very other powerful candidate here for host-directed therapy. Although we still don't know exactly what's the mechanism, we suppose that this may be happening by promoting efferocytosis of infected cells. For TB in particular, it's kind of different than from other infectious diseases. Several other intracellular pathogens, when you promote necrosis through pyroptosis, necroptosis, etc., and you have this release of pro-inflammatory mediators, you increase the immune response. In TB, it's terrible if you have necrosis. So maybe we can be preventing the development of necrosis here and inducing a more effective development of adaptive immune response. This is not very nice yet, but you can see that there are more foci of inflammation in mice that were treated only with Bago than those treated with Gas6. But now also André is working on trying to evaluate if really the promotion of efferocytosis is what's happening in these mice. And he's also starting to test if the treatment with Gas6 can lead to a host-directed therapy. The complication here is that we use purified protein. It's very, very, very expensive, and to treat mice with this is complicated. But I think we're going to have a nice result here. And so I'm going to stop here. This is what I have been doing, and just to show you, this has been done. The first part of it has been developed in Alan Scher's lab while I was at NIH. The second part that has been developed in Dr. Vania Bonato's lab while I was a young investigator in Ribeirão Preto. And now this is my group. Everybody's going to move with me here, except Gabi. Gabi is she's TT3. She's going to be in Ribeirão Preto because she's going to move to another area. She's going to study butterfly development. She's not going to study the technology. I wanted her to stay, but she's going to do other things. So thank you very much, and I'm open to questions.
Thank you very much, Diego. We open for questions, and as we normally do, we start with the students here in the room, and then at the UFRJ, and then later the more senior can ask questions like you, Magnus. So just hold on for a second, okay? Anybody here? People are shy. First lecture. This anybody from your side, Miriam?
Not right now. We're I don't see any raised hands yet.
Okay, so then let's Magnus warm up the crowd. Okay.
Well, congratulations! I'm very happy that I have you in my lab. I know that.
Yeah.
I have a question concerning the parasites. My question is how general is if you compare, for instance, with Leishmania, that is also an intracellular parasite. If they have anything that is similar to what you find in TB.
So yeah, that's what I was pointing out about necrosis and apoptosis having different roles in different intracellular bugs. So for Leishmania, if you induce apoptosis, you're going to favor infection, right? There is that beautiful work developed by David Sachs with the Trojan horse, neutrophils acting as Trojan horse. So they get apoptotic, and now he just published something with merticane and laxo receptors as well, saying that actually these receptors are important for those neutrophils to be phagocytosed and to trick macrophages to not be activated, right? And also there is a huge amount of data on iron metabolism in Leishmania that was developed by Norma Andrews in the University of Maryland. So we talked to her before we came here when we started to work on iron. And also, so Leishmania uses iron that is internalized by these cells to feed, right? But bacteria too. TB also uses iron to feed. It secretes cytophores, right? So these cytophores are going to sequester iron from the host cell, and this is used for TB maintenance and growth. But the thing is, TB, if you take TB and culture them in simple culture medium, devoid of iron, they will grow very well for 3-4 weeks. They have a massive amount of iron storage. And for different mycobacteria, iron has different roles on just the biology of the bug. For Mycobacterium avium, for example, they are heavily dependent on iron transporters that are expressed by the macrophages. For example, cells that are NRAMP1 positive. So NRAMP1 is going to feed iron to the phagosome. Mycobacterium avium grows very well. Black 6 mice, they have a mutation that they don't express functional NRAMP1, right? So they are naturally more resistant to M. avium, for example, just because they don't put iron inside the phagosome very well. But for TB, you don't see that difference. So I think the main difference is not just in feeding bug the iron here, but there must be something, well, there is something iron is doing with the host cell that is also helping. Of course, if somebody asks me, oh, are you going to target iron metabolism? No, because then you're going to screw up the whole system. but there may be very well downstream signaling cascades that can be targeted, as we saw here for
They are anemic patients. The anemic patients have a higher risk to develop tuberculosis. I think it's important.
Yeah, and actually there are very old papers from the 1800s where they treated anemic TB patients, supplemented iron, and it made everything worse. So, and this goes back to, I remember this paper is from French researcher from the 1800s, Armand Trousseau. And then there's these papers from the 1960s where in South Africa, TB is terrible. And they also were trying to supplement patients with anemia with iron. So you have dissemination, like extrapulmonary TB, you favor extrapulmonary TB. Especially in this case for the ferroportin project, we are also willing to address what happens in the liver and in the spleens, which are the main organs where you have red blood cell recycling, right? So there you really have accumulation of iron in the macrophages, and TB can grow very well in these organs if it has the proper scenario. We believe that maybe these mice will have more dissemination of TB under effect.
Just to check Miriam's side, anybody?
Now, I have a quick question, but if there are more students or postdocs over there that want to ask.
There is a couple of postdocs here, and Anderson is Sergio. Okay, Anderson, I think you first.
So thank you, Diego, for your nice presentation. Welcome to our department. I hope you have a bright future in here with us. So my question is more like conceptual, like in terms of the focus of your research, which is developing new therapies. This relationship between iron and heme proteins and the effector molecules, you showed nicely, nitric oxide. How much you can transfer that to human tuberculosis? Is that like the ROS? Can you make the same relationship? Like, do you have any results in human cells or evidence that is like connections and pathways you just showed can be, because that's the ultimate goal. So you need to make sure you can go.
It's very tricky when we talk about nitric oxide and INOS. In mice, it's beautiful, right? In humans, not so much. But there's a huge discussion about the role of INOS in humans. Well, before that, so there is a study done in vitro with human macrophages, right? Peripheral blood mononuclear cells that were differentiated into macrophages, and these guys infected those macrophages, treated with the same inhibitor. There's a decrease in bacterial loads. So it works in human macrophages. It's hard to treat humans with this drug, right? This drug was used in humans for treatment of jaundice in newborns, because it's going to inhibit the degradation of heme, right? But this drug has some toxicities that are not very bad, but you have you become photosensitive. So if you inject timperoporphyrin in a person, they need to be kept away from the sun for some hours. Otherwise, they're going to have burns in their skin. So it's complicated. There is a company that developed an H1 inhibitor, Mallinckrodt, but I think it went bankruptcy. But those guys sent us the drug, and we were testing together with some people in TB Alliance. So we're proposing to do this with other mycobacteria as well. But so inhibiting H1 with this drug in human cells works. Is it through INOS? I don't know. Especially when you talk about peripheral blood monocytes that were differentiated into macrophages. They don't express INOS. But there is this work from Carl Nathan, right, from Cornell. And he was the first one that demonstrated that INOS knockout mice are extremely susceptible to TB. And he continues to believe firmly to this day that if you leave peripheral blood out of the question and go to the lung and examine the expression of INOS there, you're going to find it. And actually, there is another paper in HO1, but the guy thinks differently. Actually, not so differently. If I treat mice for 3 weeks with HO1 inhibitor, it's okay. If I continue to treat it forever, then it's going to be bad because you have ROS production, heme is toxic. We all know that. But it's something that you can have a very acute intervention that will help. This guy from South Africa, Adrian Steen, he has several samples of patients that were not responding to treatment. You have to do lung detection. So he stained beautifully iNOS expression in the macrophages that are in the granule. And very nicely, where he sees HO1, he doesn't see iNOS. Where he sees iNOS, he doesn't see HO1. But he is more to decide, oh, HO1 is good. It is good, it's cell-protective. But maybe if you give it a burst in the beginning, you can have the macrophage work better, then you stop and let the organ to do its job.
Miriam, let's move to your side and then back here later.
Okay. I'll ask a quick question, based on my ignorance, actually, Diogo. You showed very nicely the difference between the HIF1 alpha effect and the HIF2 alpha effect. And I wonder if it have been shown that the HIF2 alpha can maybe work as a homodimer, or if they would have other partners, or if you think it's just the absence of one that would impact the functionality of the other one, or something like that. Like look back at the NFAT studies, right. and there are some strange results, and then you saw that in some sites they can bind as homodimers, in other sites they need partners, and then there's the difference of the genes that are being activated that make a difference in the response. So I wonder if you know anything about that for HIF.
So it's great to hear you suggest that, because I never read those papers and I need to read them now. I didn't know they could act differently if they were monomers or dimers. But in the beginning, we thought that it would be merely something about which phenotype of macrophage is being induced by each one of them. It seems that's not the case, because we are not seeing too much difference in iNOS or Arg1. So we did some experiments measuring arginase 1 activity as well, and we did not see a difference. But it's interesting that you have this higher expression of HIF1 alpha when you don't have HIF2 alpha. We don't know if this is merely compensatory, or if there is really some role for HIF2 and HIF1 to counter-regulate each other.
Mm-hmm.
And definitely, I don't know if there is something related to them being expressed as dimers or monomers. It can affect. That's definitely something I need to study and to think about. Thank you.
Mm-hmm. Thank you. Thank you.
Anybody else from your side, Miriam?
Maybe Julio just opened his camera. Julio, do you want to ask a question?
Yes, such a wonderful, dense presentation. Congratulations. I have a very basic question, which has to do, I think that mostly early stages of infection, when you don't have so much tissue destruction, what are the major sources of the iron? Do you think, is it transferrin that has to reach the infection site in the lung in order to deliver the iron? I would suppose that the iron has to be, transferrin has to be processed in order to release the iron for the intracellular transport, right? Do you think that ferritin could also play that role? And the second question, which has nothing to do with this, is related to ADAM17. Do you think adaptive responses, TH1 responses, do you know if there is modulation of ADAM17 in tissues? And what is known about the autopsies of people that have died with tuberculosis? If there is autopsy information about the tissue distribution of the myriad of factors that you have discussed in the late stages of your presentation.
Right. Yeah, so about iron, like first question. What has been shown so far for iron accumulation in lung tissues from people with TB? You already have like tissue destruction, right? So they find a lot of iron in the necrotic tissue. In the initial stages, what I think might be happening mice is that cells are going to rely more on the internalization of transferrin, as you suggested. I think that's what happens. And actually, that's why I think the effects of H1 inhibition only work when you have establishment of infection 3 weeks post-infection, right?
Right.
And then you have a very, I didn't show, but also the induction of H1 expression occurs together with the induction of interferon gamma production and everything. So I think there is, not just I think, there is a role for pro-inflammatory cytokines, interferon gamma and TNF among them, in inducing H1 expression. And there, these macrophages can be recycling hemoproteins inside the cells. They can be, I tried to stain for intracellular red blood cells in lung macrophages from TB infected mice. You can find some, but it's not that much. So I don't think they are phagocytosing that much red blood cells in the lungs. They may be feeding, I would believe, mostly of hemoproteins on that point. And also, I think transferrin receptor is an important source of iron for those macrophages. Actually, a part of my students' PhD thesis now, one of the objectives is actually to deplete DMT1 in macrophages and see if we can have modulation of the, we're believing that this is happening through ferroptosis. So we're gonna see if the necrotic lesions are lower when the mice do not have DMT1. But it's tricky to suggest if it's happening through transferring or hemoxygenase in each phase. I would believe it happens as you suggested. Maybe mainly in the first phases, transferring is more important, and later H1. Regarding ADAM17, I truly have no information about how it's modulated in TB. And I know it is induced in response to several pro-inflammatory stimuli, LPS, ROS, and pro-inflammatory cytokines, but I don't have information on how it works in TB. It's definitely something that we want to evaluate as well.
Thanks a lot.
Sérgio,
Yeah, congratulations for the talk. Have you tried to drive M1 macrophages and supplement this iron and see reverse to M2 and see maybe a stabilization of H2 alpha?
I did not try that. But it's something that we're willing to do. Actually, we did not try to drive them towards the M1 or M2 yet. What we're trying is to see if iron can enhance the markers for M1 and M2. This is something Marine is doing, but this is a very nice idea to try to as well.
Motilo,
What about the natural resistant gene that people have shown that's very important in mice for tuberculosis, as well as in Leishmania
Right.
and in Salmonella typhimurium and so on?
So that's the thing. So NRAM is very important for some mycobacterial species. For avium is extremely important. They said just the fact of not having a functional natural resistant gene NRAM1 expression, the mice get more resistant, right? This DMT1, its original name is actually NRAM2. So it's the natural resistant gene too. But it does not work the same way as NRAM1, right? Actually, I think there is not a consensus about if NRAM is pumping iron into the phagosome or out, or if it does both ways. At least I think most of the evidences point to it pumping iron inside the phagosome. And as I said, for several mycobacterial species, it's extremely important, mainly for the non-pathogenic ones, those that affect mostly people that are immunocompromised, the non-tuberculous mycobacteria. If you have a defect in NRAM1, actually it's good. You're going to be more resistant. But in TB, that doesn't happen. So there is a paper where they use the 129 mice that have NRAM1, functional NRAM1, and black 6 with TB. They're equally susceptible. Then they isolated the mutation, transferred those to 129, and it did not make a difference. So, as I said, what is known is that TB has very good iron, very nice iron storage that can resist this initial lower availability of iron, let's say like that. So that's why I believe for TB, the effects of iron must be more important in modulating the host cells than the environment where the bacteria is growing itself.
All right, guys, I think we're running very late. Bruno and I has another course to attend to. So I'd like to thank very much Diego and all the audience, and see you next Monday. Thank you very much.
Thank you very much, Diego, and thank you for an amazing talk.
Valeu.